So I want to talk about GWAS, or Genome-wide Association Studies, and they're a really important biological tool in genetics to determine what genes are responsible for particular traits or disease states, and they're really interesting, and hopefully soon everyone will be able to get their genotype read out when they go to the doctor, and GWAS will be able to give us some really good information on how patients respond to particular drugs and particular interventions. So getting started here, I would say Francis Galton's kind of the intellectual author of genome-wide association studies. He was the cousin of Charles Darwin, and he had a kind of a pet project of showing how traits are heritable from one generation to the next. And it turns out that they are, but he was puzzled by a couple things. One being that, very tall parents produced shorter offspring than themselves. So that was kind of weird. And he found a statistical rule that followed that determined how exactly traits were heritable. And there indeed seems to be some kind of mean that all traits tend towards, but he never really found any interesting metrics to use. He tried to be able to predict behavior, specifically things like IQ, but he never really found any sound methodology to predict behavior. So like I said, GWAS allowed us to take a trait or take a disease state and let us form a correlation between particular genes in a population and the occurrence of that trait or disease. So that's the first step. You have to sample and collect the sequencing, and then you're going to do data analysis, and we'll talk about how the data analysis for GWAS look in a minute. But I'll be talking about SNPs. SNPs are just single nucleotide polymorphisms, just genes that vary in a population. There's no reason to look for genes that don't vary. Those are not interesting for any kind of variability in a population, which is important for heritability. So the reason for the big waves that GWAS are making in behavioral genetics is because mostly psychological studies are pretty not great. A lot of people know this, but if you look at 100 psychological papers, only 36% around will replicate or yield significant results. On the other hand, behavioral genetics has not been worried about this. They've been charging ahead and coming up with good amounts of variance and results in spite of all the bad psychological studies. And this is confirmed in a number of ways, the GWAS themselves, and through twin studies, we've known that genes are playing a big role in heritability. So what heritability is exactly? Heritability is a score that determines the genetic role and variability of a trait. It only describes what is in a population. It's not describing what environmental factors are making the trait that variable. It's just describing the gene's role in determining the variability. And it doesn't reflect a single individual, but individual differences at the population at large. And that is in a certain time and in a certain environment, obviously, but GWAS don't really control for the environment in most studies. They're mostly just looking at what are the genes doing in this environment? What are the genes doing at this particular point in time? It's not describing what is possible in a population. So since I've said that once, let me just reiterate this. So how much do genes have to do with the average level of this trait? This is not what heritability is. This is what a lot of lay people come away from the field thinking. This is not that. A way to kind of exorcise that thought is to think about the heritability of number of fingers. You would think that it's 100% right. Genes have a lot to do with the number of fingers that you have. Hox genes are going to determine the gross body structures of the body. But unintuitively, remember this is a definition we're using. This is not an exact descriptor of the world. It's 0%, even though genes have a huge role in determining the number of fingers. what heritability is what do genes have to do with the degree of variability around the average of this trait. So the variability, how much can this gene explain why someone has 9 fingers instead of 10? All likelihood, probably not a whole lot. The takeaway is if you're trying to know why someone has 9 fingers instead of 10, knowing that they juggle chainsaws at the carnival on the weekends is going to be more important than knowing their genotype. So in going about talking about this, we need to ask what role does this variable or this gene have to do with the variance, the reason that people differ. So looking at this graph, we can kind of get an idea of what kinds of genes GWAS are looking for. So on the x-axis here, we have allele frequency, just how common these genes are. And up here on the y-axis, we have effect size. So within this box is all the possible combinations of genes that could be possible. So starting out in the bottom left corner here, we have rare variants with small effects. These are genes that they're very rare in the population. Not many people have them. And they have low effects they don't describe much of the variability or reason why people have a disease or don't have a disease or have a trait and don't have a trait. So these are not really interesting to look for with GWAS. On the other hand, common variants with small effects, so just moving right here, common variants, most people have them, low effect, not very descriptive, at least individually, in describing why someone has, say, prostate cancer. But diseases like this, if you have enough participants, GWAS can really find these associations very well. Conversely, something more difficult or maybe not even interesting to find with a GWAS is common variants with large effects. These are things like Alzheimer's, BRCA1, BRCA2 genes for breast cancer. These are the kinds of things that genetic counselors can just take your blood, genotype you, and they can see with their eye that you have this gene or that gene, and that there's a huge potential for you developing that disease, that trait. So not really useful for genome-wide association studies. Cystic fibrosis, also something that has a very high effect, these genes that contribute to cystic fibrosis, but they have a pretty low penetrance, so they're pretty rare. These are not particularly easy to measure with GWAS. It can be done, but what we're mostly looking for is these two right here. And the one we haven't talked about is less common variants with moderate effects, Crohn's disease, type 2 diabetes, and we'll talk about how studies have been using GWAS to explain variants in type 2 diabetes in a minute. But these are the butter zone right here. These are It's not easy to find these kinds of diseases or traits because they have lower mounted effects. So there's many genes contributing to this phenotype. So what are GWAS anyway? What's kind of the mathematics behind it? They mostly deal with additive models. So if you have this gene, it will explain an additive proportion of the variance. So we say we have this gene right here. This capital A right here, you have this. It should be hypothesized if you're predicting that this allele is going to explain some of the variance, that having one copy will give you an effect, and you can measure it among people who don't have this. And then you can also predict that the exact same effect will be additive if you have a second copy of this gene. So that's not always how genes work. There is multiplicative and nonlinear interactions where if you get a gene, this set of 5 genes, you'll have a huge propensity to exhibit this disease. But these are very computationally hard to look for, so GWAS pick on the low-hanging fruit. So given the frequency of genetic variation among a population, what is the incidence of a phenotype among participants with and without this SNP or polymorphism or gene? There's 2 ways you can really look at it. You can ask, what are the odds of gene A being present when the disease is present? Okay. What is the odds of gene A being not present when the disease is not present? So there's two ways to kind of look at it there. When the gene is there, it just so happens that people who have this gene are more likely to develop this disease. So, and conversely, when they don't have this gene, they're less likely to develop this disease. Pretty straightforward. I won't get into the math, but genome-wide association studies yield these Manhattan plots, which are fun to look at. So on the x-axis here, you have the chromosomes, and individual dots, they're almost too tiny to see, are genes, individual genes on the chromosomes. And over here on the y-axis, you have effect size. So what you're basically looking at, most of the colored regions below my cursor are genes that aren't very interesting. Everybody has them, and they're not really doing anything. But genes above my cursor here, and this dashed line, these genes are significant because people who have these genes, these are clusters of genes here, these are many genes within this dot, people who have them are correlated with the trait in question or the disease in question you're looking for. And this is merely correlational without further investigation. What you'd want to do after you find the genes in question, these genes, you would need to run animal models on them. Knock this gene out and see what it does in an animal model. Does it have a relationship to the disease or trait in question? So with that, some of the main findings that GWAS have been showing, and twin studies as well, all psychological traits are substantially genetically influenced. Broadly speaking, all traits around 30% to 80% of the heritability can be described by the genes. And we know this because in terms of simple twin studies, we see that all the time, identical twins are more similar than fraternal twins. And it's not because of the environment. In diseases even, like schizophrenia, we see a higher concordance in identical twins than fraternal. Even more hard metrics to measure, such as IQ, hovers around 50% heritable, and personalities 30 to 50%, and even really environmentally dependent measures like well-being are coming in at 38%. So the takeaway, and just to remind you guys, a score of 100% heritability means no current environmental factors are changing the likelihood of inheriting this trait or this gene. Which takes me to the next slide. No traits are really 100% heritable. There's some that are, if you have this gene, you're going to get this disease, but in traits, neuroticism, extroversion, stuff like that, it's not one or two genes, it's thousands or even more, and they're all contributing to this trait or disease. And this is what Galton kind of wondered about. If two 7-foot tall parents had a child, what's the likelihood of that child being 7 foot tall or taller? So say you and your spouse were out here on the 95th percentile for height. The rest of the population's to your left or shorter. What's the likelihood that your child is going to be taller than you or just as tall? I'll tell you what it is. It's a hell of a lot less likely. It's less likely for your child to be taller or as tall as you than it is for them to be as tall as you or shorter. This is just a statistical necessity because more of the bell curve is on the left side of where your parents are. This is regression to the mean. No matter what, in later generations, things tend to go towards the mean. Even if you start with two very tall parents, the likelihood, and this goes for any trait tends towards this mean, tends towards the population average, no matter where you are on the curve. So this is why kind of Galton was perplexed, but this is also why Galton was wrong in terms of, having this very high-minded idea of IQ and heritability. Genius is a very rare thing, and it's, occurring out here on this very slim tail, and, even if two geniuses get together and have a kid, the likelihood that they will be less bright than their parents is just enormous. So it's really just lightning in a bottle, really. So for anyone that wants to calculate a polygenic score on any trait or disease, you know, we're living in a day and age when you can get your genotype done, and you can count, if you know the genes that you're looking for, you can add up your polygenic score for anything. And how you do that is, say this is these are the disease alleles right here, and you've identified 10 S&P locations that you're looking at. So you have an increasing allele right here. Let's say we're looking for risk of attending a boring business school. So you want to know the risk of you attending a boring business school. This is the gene that is associated with that trait. And looking at this S&P, we see that you have an A in this location, a non-disease allele, and then we have a disease allele here. That is going to give you a genotypic score in this S&P of 1 out of 2. So we have to multiply that with the correlation with the trait. How correlated is this particular allele with the trait in question? And you can find that out. And you multiply your genotypic score with the correlation of the trait to get a weighted genotypic score. You can do that with every S&P and get a polygenic score that can tell you the odds of you exhibiting that disease or exhibiting that trait. So heritability is caused by many genes of small effect. We've been over this a little bit, but just give you an idea of how many participants you really need to have to see this. A GWAS of 20,000 participants has a 99 almost perfect % accuracy to detect an association with an effect size that accounts for 1% of the variance. But they just don't do that. And the reason for that is that genes are contributing very minuscule amounts of the variance per gene. So you need to find tons and tons of genes of very low effect to really find out why this person is exhibiting that phenotype. So how we know this, there's animal models here. So down here on the x-axis we have generations, and on the y-axis we have this behavior. Call it its open field activity, but it's really a measure of anxiety in mice. So you have these control lines right here that are not selectively bred for this phenotype, and you have this control line here that it's positively selected for that phenotype, and then you have lines here that are negatively selected for that phenotype. So in both cases, you can breed mice to be very gregarious, very unexcitable, they'll just go to the middle of whatever box they're in with no fear. Just like the dodo on the Galapagos Island, it kind of just hung out and had no fear of any invaders. And then we have these shuttered mice down here, the Edgar Allan Poe types. So what we see is the reason we know that many genes are contributing to traits is because every generation we're seeing differences in behavior, and as we're going up with the lines and selectively breeding them for this behavior, we're seeing an increase every generation. What that means is that through the selective breeding, they're additively getting more traits for this behavior over the generations. And it's not just one big inheritance event. If it were the case that 3 genes contributed to this phenotype, we would see 3 distinct bumps, and there would be very little ups and downs, or there would be very distinct up and downs. But it's not the case. We go down or up in this very hitchy, up and down, stair-steppy manner, and we can see that sometimes they get genes for this trait, sometimes they don't, but it's going up and down consistently. So the shuffling around of traits is, the shuffling around of genes is determining the effect, the phenotype here. So top 10 clinically effective, top 10 most lucrative drugs in the US have a clinical efficacy, meaning that they do what you want them to in the degree that you want them to, is 1 in 4. So top 10 drugs work 1 in 4 times. Why is that? Mostly probably because the genotypes in question are not assayed. You can get your genotype done sometimes at the doctor's office if they're looking for a particular variant and they know what they're looking for, but most of the time genotypes are not integrated into healthcare, which is something that needs to be done to help make up this gap in pharmacological efficacy. So for things like diabetes, which we talked about, where the genes have a moderate effect of explaining why someone has diabetes, we need lots of participants and patients to actually use a GWAS to find this effect. How many? Almost 1 million, which is quite large. And the study that collected this data used many studies. So what we get with that is that we have 243 loci, which is a lot of genes in question. And with those loci, we explain 18% of the variance. So 18% of the reason why someone does or does not have diabetes can be explained by these 243 loci. So in my area of research, something like even neuropathic pain has come away as being something that's fairly heritable. About 1/3 of the variance can be accounted for by genes, by twin studies. There's several problems with classifying chronic pain, but GWAS recently have found a lot of prospective genes, and the reason they're so good, GWAS, at finding the minute associations is because most people don't look for metabolism genes and immune response genes when they're talking about chronic pain. They're looking at the neurotransmission and ion channel genes. So these let you look for associations with no pre-configured hypothesis. So you're not looking over here, you're looking at where the genes are leading you. So how big are the variances we talked about? How big is 18% in the case of diabetes, or 38% in terms of neuropathic pain? Well, sex can explain about 1% of the phenotypic variance in most traits. Depends on what you're looking at, but something, just to give you context, something explaining 10% of the variance is going to land any psychologist in any institution that they really want to land into. Frequently, psychological journals are publishing papers that are explaining single-digit variances of some variable or the other. With these genotypic effects, we're seeing that we can explain double-digit variances pretty easily with these GWAs. Some limitations for genome-wide association study is occurrence. Thing that is done is to look at a gene, see if it contributes to explaining a percent of the heritability, and throw it into the analysis and say that this gene is correlated with this phenotype or this trait or this disease. There's ways in which that can really hinder or at least give the perception that you're not really looking for a causative explanation. For instance, if you're looking at a novel population and you see that a couple genes are novel in the population and you're trying to explain a phenotype, so where does the field go after this? After whole genome analysis is used, which is a computational nightmare, there's other proteomic, which is what proteins are being expressed, transcriptomics, what transcripts are being expressed. Those things can be integrated, but they present such a computational nightmare that whole genome is really the best we can hope for in the near future. Multiomics is promising, but quite a ways away. But correlating these genes with particular traits is going to be a big first step. So I'll leave you with this quote from Moby Dick that I stumbled across, and I found it interesting that Melville thought this way, considering if you look at the graph over here, we can see that most of these traits are falling into a nice bell curve, and 80% of the population is within the first standard deviation of one side of the mean or the other. It's just really profound that we're all so close. We've been talking about dissimilarity the entire time, and it's just nice to take a step back and wonder why we're so similar.